So, today I'll talk a little bit more about DNA replication and the end replication problem, and in particular, the role of the enzyme telomerase in telomere formation and maintenance. So, when we think of DNA replication, of course, we think of these replication bubbles formed at origins of replication, where double-stranded DNA is opened and each of the strands is used as a template with DNA polymerase to generate a new daughter strand of DNA. Eventually, these bubbles will run into one another, and where the bubbles fuse together, then you'll just end up with parallel identical chromosomes, and eventually these will be the daughter chromosomes that form during mitosis. So let's look at the details of DNA polymerase and replication in a little bit more resolution to try to think about some of the problems that cells run into with the process. So like all enzymes, DNA polymerase is very, very particular about the reactions it catalyzes. And in fact, DNA Pol specifically adds new nucleotides to an existing 3' end of a DNA strand. So if we label up the particular ends of the parental or template strand and then the new daughter strand, you see that for this particular new strand that's forming, there's a little RNA primer in red that DNA polymerase can extend in the 5' to 3' direction by adding new nucleotides to that 3' end. So interestingly, DNA Pol can add to an RNA primer or can add to an existing 3' end of a DNA molecule. So it's very particular about direction and orientation, but not so much about the type of nucleic acid it's extending, which is interesting. So as time goes on, this so-called replication fork opens up, exposing more single-stranded template DNA, and DNA Pol just continues zipping along and extending that strand. So it turns out this is called the leading strand in a replication fork, and that strand is synthesized continuously in one big long DNA molecule from 5' to 3' as the fork opens more and more and more until it eventually runs into the next replication bubble. If we consider what happens as we go through the mechanics of this process of DNA replication, we can start to see a problem emerge. So, as I mentioned before, DNA Pol will extend from the 3' end of an existing nucleic acid that's bonded by complementary base pairs to a template. So here we have a little primer, and that little primer was actually formed by a type of RNA polymerase called primase. This is an important distinction between RNA polymerases and DNA polymerases, and that is RNA polymerases can initiate a new strand, whereas DNA polymerases require a 3' end of an existing nucleic acid, either RNA or DNA, to extend a strand. So DNA Pol by itself can't initiate a new strand. We need to have primase to initiate that new strand, and then DNA Pol will only extend that strand. So if we now consider the fact that we have linear chromosomes rather than circular chromosomes, you can imagine that at the end of a chromosome, we're going to run into a problem. So in the top strand in this diagram, the pink strand, notice the orientation of synthesis. As RNA primers are added by primase, DNA Pol can extend from 5' to 3' to the right, in this particular case, on that top parental strand. And so DNA Pol can synthesize a complementary sequence all the way to the end of the chromosome. No problem there. On the other strand, which turns out to be the leading strand, the RNA Pol called primase doesn't actually place an RNA primer at the extreme end of that blue parental strand. In fact, it might be 50 or 100 or 200 nucleotides short of the extreme 3' end of that particular strand. So now, as DNA synthesis proceeds, we'll actually lose that particular bit of template the next time DNA replication occurs, because now this bottom strand template will eventually lead to the formation of a new daughter chromosome, but that 5' end will be a little bit short. So in the next round of DNA replication, when we come back the opposite direction, we just won't have that little last 150 base pairs on that template that has the 5' end that's paired to the blue parental strand down in the bottom. So we lose sequences from the end of the chromosome every time they're replicated, because primase doesn't place a primer right at the extreme end of that bottom 3' template. So this is called the end replication problem, and it leads to loss of sequences. Fortunately, those sequences are just these repetitive telomere sequences, and as long as cells don't divide more than 80 or so times, there's enough telomere to deal with this loss. However, if cells proliferate enough times to degrade those telomeres down to about 3 kilobase nubs, that activates this whole senescence or even crisis program to prevent cell proliferation. It turns out that, interestingly, a lot of what we understand about telomeres was learned in various aquatic ciliates. So you've probably heard of Tetrahymena, and there are others. One of the cool things about these ciliates is that they have some unusual nuclear structures. Some of them have both a macronucleus and what is known as a micronucleus, and there's some actually crazy chromosomal gymnastics that results in shattering or cutting up the chromosomes into tiny little microchromosomes, each of which has its own little telomeres. So it turns out that these particular critters tend to have a lot of telomerase and a lot of telomere forming potential. So a ton of what we know about telomere biology was actually learned from these critters. And in fact, the folks, including Elizabeth Blackburn at UCSF, who did a lot of this work on these ciliates and their telomerase, were awarded the Nobel Prize for their work. So these funny little pond organisms have been really important for understanding telomere biology. The particular enzyme required for telomere formation is called telomerase. And telomerase is interesting. So telomerase is a ribonucleoprotein, so it has a protein component, and indeed the catalytic component is a protein enzyme. So it's you can think of telomerase as a kind of DNA polymerase, but interestingly, it has an RNA subunit, and the protein component is called TERT. In humans, it's H-TERT, and the RNA component is called TR. So that's the telomerase RNA subunit, or HTR in humans. So the complete enzyme, the holoenzyme called telomerase, has both that protein and the RNA subunit. If we actually look in a little more detail at that RNA subunit, it has a complex secondary and tertiary structure, and ultimately the important bit about telomerase is that there's a loop of telomerase RNA sequence that serves as a template for the DNA polymerase function of telomerase. So telomerase is an enzyme that uses an RNA template to synthesize DNA. Fascinating. So here you can see using its own built-in template, it's added another little hexanucleotide sequence to the 3' end of that strand at the end of the enzyme. So the holoenzyme is required, both the RNA and protein subunits, in order for this activity to be present. And in fact, when one monitors human tumor samples, the vast majority of those samples have telomerase activity. So most tumors have the ability to extend their telomeres. And this is quite unusual compared to somatic cells because somatic cells do not have telomerase function. It turns out in those somatic cells, the RNA subunit, which is the template for telomere extension, is not present. On the flip side, germ cells, which are the cells that give rise to eggs and sperm, and then it turns out stem cells actually do have telomerase activity. They do express both TERT and TR. So now if we think of cancers as perhaps diseases, proliferative diseases of stem cells, it might make sense that cancers are very likely to have telomerase expression if indeed they are derived from stem cells. So if we think about the fact that telomerase, the TERT subunit uses the tRNA subunit as a template, that actually describes a particular kind of DNA polymerase called a reverse transcriptase. And we've talked about reverse transcriptases before. Classically, these had been known as enzymes used by retroviruses to convert an RNA retrovirus genome into a DNA provirus that is then inserted into the host genome. But if you look at the gene structure of viral reverse transcriptases and mammalian or other organismal telomerases, the domain structures and the overall structures of these proteins are really quite similar. So they are very likely evolutionarily related, and probably various retroviruses picked up telomerase, and it eventually evolved into reverse transcriptase. Or it quite frankly could be the other way around. I'm not really sure what the current state of the art on that thinking is. So telomerase is a really important component of maintaining the telomere length in cells, and lack of telomerase activity results in cells that are essentially mortal. However, if telomerase is active, then cells are functionally immortal in many cases. So it turns out that if you take human embryonic kidney cells and grow them in culture for a long period of time, they will eventually undergo senescence or transition into crisis and commit apoptosis because the telomeres have eroded down to a critical length. If, however, you force expression of the protein subunit, TERT, this allows cells to not undergo senescence and crisis and allows the cells to continue to proliferate. So it seems that expression of TERT and allowing telomerase to function is sufficient to functionally immortalize these cells. So if you actually look at a variety of cancer cell lines that have TERT expression, adding more wild-type TERT does not affect their ability to proliferate in cells. However, if you can introduce a form of TERT that has a dominant negative activity, so imagine a TERT that is catalytically inactive, that perhaps can't make telomeres, but can still bind to the TR subunit, the RNA subunit that is expressed in these cells, then you might be able to sop up the RNA subunit so that telomeres can't be extended. And in fact, when that occurs in this cell line that has very short telomeres, they actually undergo senescence and or crisis very rapidly. If you repeat the experiments with cell lines that have somewhat longer telomeres, there's a little bit of a delay, a little bit of lag between the introduction of the dominant negative TERT and induction of senescence or crisis, but eventually, once these cell lines erode their telomeres down to a critical length, they too undergo senescence and or crisis. So it really does seem that telomere length is a determining factor, and therefore telomerase activity is a determining factor in the immortal behaviors of cancer cell lines and other normal healthy cells like stem cells. As you might expect then, uh, telomerase activity is a good prognostic feature when examining various cancers. So tumors that don't have telomerase activity in the blue traces on these Kaplan-Meier plots have very, very good prognosis and low levels of relapse, whereas individuals who have TA positive or telomerase activity positive cancers have a much, much decreased likelihood of survival. So telomerase activity is a very, very good prognostic marker for the likelihood of relapse for a whole variety of cancers. I mentioned, however, that only about 90% of human cancers have telomerase activity. So there's another 10 or 15% of cancers that actually don't seem to be telomerase positive. And this is sort of an interesting situation. If those cells didn't have some way to protect the ends of their chromosomes, you would expect that essential genes would be eroded from those chromosomes, leading to cell death. So it turns out that there are alternative mechanisms known as alternative lengthening of telomeres mechanisms, that can actually copy telomeric sequences from one chromosome to another. As we'll see, we can think of this sort of being akin to template switching or sort of gene conversion, but right at the end of a chromosome that can allow for the extension of terminal sequences. So you can imagine that the free and the single-stranded region of the extreme end of a chromosome, the telomere sequence, might be available to actually hybridize with a complementary telomeric region from another chromosome, and this is called an interchromosomal copy choice mechanism. So DNA Pol can effectively use another chromosome, since the telomere sequences are homologous to one another, and that second chromosome can serve as a template to extend the telomere of the second chromosome. So effectively, there's a mechanism to use regular DNA polymerase rather than using telomerase to extend the template. So you can imagine a primase can come in, generate primers, and then DNA Pol can synthesize the complementary strand of that strand that had been extended by this copy choice mechanism. So again, sort of like an efficient version of template switching targeted to the ends of chromosomes. So this alt mechanism seems to be present in those cancers that lack telomerase activity. And understanding the mechanisms that control this copy choice, interchromosomal copy choice, will be very important for thinking about therapies for those cancers. Obviously, telomerase is a potentially interesting target for chemotherapies, although because its activity is required in germ cells and in stem cells, it's a little bit of a sticky wicket. It's interesting that when one monitors telomeres in mice, they're much, much longer than those in humans. And when you think about the life and times of a mouse, mice are quite different than humans. They have a very short lifespan, and they don't need nearly the proliferative capacity in their cells that humans do. I've also mentioned that mouse cells are much easier to transform than human cells because, again, for the most part, cancers are a problem of long-lived organisms like us, and if you only live for a couple years anyway, the likelihood that cancer will be the end of you is quite low. So these organisms don't really use telomeres in the same way that we do as part of the timing mechanism to ensure cellular mortality. So in fact, if mice lose telomerase activity, one will see that their telomeres shorten about 5 kilobases a year, but it's really not until the 5th or 6th generation that one starts to see phenotypes associated with these shortened chromosomes. So basically, organisms with unusually short chromosomes tend to exhibit phenotypes that we often associate with old age. Decreased proliferative ability in cells corresponds to decreased wound healing and just a general lack of vigor. Particularly in tissues that are highly proliferative in healthy organisms, we see major deficits. So the epidermis that has a lot of cell turnover and the epithelial lining of the gut has a lot of cell turnover, and those particular tissues are pretty hard hit in these organisms. So as you would expect, they're very feeble and not healthy at all. So finally, if one actually monitors the length of telomeres in various cell types over the years, you can see that in these various circulating blood cell populations, that as we age, our telomeres get shorter and shorter and shorter, starting off at 11 or 12 or 13 kilobases, and by the time we're 80 or 90 or 100, they're getting down to just a few kilobases and really reaching that critical level that can induce senescence or crisis. So the shortening of the telomeres is sort of our built-in proliferation limiting clock to ensure that cells can't divide so many times that they could give rise to a huge tumor. So the downside to this is that we have a finite lifespan of a little less than a century. But the good news is, because of these mechanisms, cancers are much more rare than one might otherwise predict if we didn't have these sort of systems. If cells acquire the ability to extend their telomeres, then they are much more likely to be transformed, or thought of another way. That's one of the very strongly selected for phenotypes in cells during transformation is acquisition of the ability to express TERT, the protein subunit of telomerase, in order to become immortal. So that's one very important step of immortalization for cells.